Thank you very much for a nice introduction. So before I begin my presentation, I would like to thank the organizing committee who actually invited me here. I'm really thankful for them. Thank you very much. So today I will going to discuss the research project. Actually, I will discuss the PhD project of my student, Dr. Rukhsana Nawaz. She worked on schizophrenia. She has identified the factors that is responsible in the onset of schizophrenia. So I'll begin. First, I'll give you a little brief of what is actually about and why we have done this. So schizophrenia is a neuropsychotic disorder which is affecting at least 1.5% total population of Pakistan is affecting from this disease. There are several type of many underlying causes of schizophrenia, not just one. And only in Pakistani hospitals, the clinical doctors or psychiatrists, they just have drugs or give drugs on the basis of the symptoms. However, genotyping for specific mutations, gene-to-gene interactions and interactions of genes to environment linked studies have not been investigated up till now. So for the first time here, we have produced a database about the factors that could lead to the schizophrenia in Pakistani population. Schizophrenia is previously misdiagnosed or didn't get full attention in Pakistan actually because due to lack of severe awareness, and it is because it is actually hereditary as well, so it transmitted generation to generation. Therefore, we have attempted to identify the factors that are involved in the onset of schizophrenia. The main problem of having this type of disease and due to the lack of awareness and the drugs because of this having side effects. So people, because as we do not have mutation-based drugs or drugs that are responsible for specific type of symptoms, so only few different multiple type of drugs has been given to these patients, which actually have a severe side effects because these drugs does not only bind to one receptor, but they also binds to other receptors. As you all know, schizophrenia is in which dopamine concentration becomes high. So it's actually the less or reduced activation of D1 dopamine receptors, overstimulation of D2 or dopamine receptors, and less activation of GABAergic receptors takes place. So usually and mainly the drugs that has been used overall is haloperidol and chlorpromazine. So as if you are a medical student or you have a biological background, you know they are neuroleptics. They tend to decrease the dopamine levels in the brain. But these drugs do not bind to only D1, D2 receptors, but they also bind to D3, D4 of dopamine receptors. Simultaneously, they also bind to serotonin receptors as well as histamine receptors, which actually gives heavy side effects and it worsens the symptoms of schizophrenia. So that's why usually many patients, they left the medication in between their medical trials and it just keep on going. So it's a miserable condition. So that's why our study will assess in the our study will assess in the broader context of enhancing the contribution of science and technology to human development, both locally and globally. So as I've mentioned, that schizophrenia is a psychotic disorder. The main symptoms are delusions and hallucinations. They also have withdrawal or lack of social environment and mood disorders. And approximately 1% of total population, world population is suffering from this disease. Obviously, as it is a hereditary disease, so the parents, obviously, children, and the first cousins, first relatives, sisters, brothers, they are definitely at high risk, as well as second cousin, uncles, and aunties could also have schizophrenia, from where you can also inherit the disease. There are several types of schizophrenia, not just one. So one is the paranoid, hebephrenic, catatonic, and residual, and undifferentiated. So these all are the types of schizophrenia that is present in Pakistan and all over the world. The common symptom is the one is the positive, negative, and cognitive symptoms in the clinical trial and the psychiatric terms. And positive symptoms includes delusion, hallucinations, abnormal movement, whereas negative symptoms involves social withdrawal, lack of movement, and mood swings and feeling sad. Cognitive symptoms in which the cognitive memory or the working memory is disturbed. So it's not just only one symptom, but 3 symptoms are involved. Usually it is said that positive symptoms are related to the overstimulation of D2 dopamine receptors. Negative symptoms are related to the reduced activation of D1 receptors. And obviously cognitive symptoms are obviously involved in the reduced innovation of GABAergic and excitatory neurotransmitter glutamatergic input in the prefrontal cortex, which is actually involved in the working memory. So usually in Pakistan, the criteria for diagnosis of schizophrenia is these 2 type of classification. So it's ICD-10 and DSM-4. These are the 2 psychiatric books. On the basis of these symptoms, the psychiatrist actually diagnose that the person is actually having schizophrenia, or is she or he is a schizophrenic or not. So as I mentioned, that it is hereditary, environmental, and anything that if brain structure changes could lead to the onset of schizophrenia. There are 40 genes have been identified up till now that shows that they have involvement in the schizophrenia. My student in her PhD work, she has identified the SNP mutation or single nucleotide polymorphism in these 3 genes: NRG1, AKT1, and COMT gene. NRG1 is neurolegulan gene, AKT1 is serine-threonine kinase enzyme, and COMT is catecholamine methyl transferase enzyme. We'll discuss it in a little bit brief afterwards. She also identified the effect of environment in the onset of schizophrenia, like metals and like drug abuse, or the effect of what you call marital status and salary bracket. So she has identified the socio-economic factors that could lead to schizophrenia, the hereditary factors that could lead to schizophrenia, and obviously if any changes in the brain structure that is present. So usually in schizophrenia, we usually seen large ventricles. They have reduced regional cerebral volumes, and obviously they have reduced activity in the temporal lobe. So actually they have auditory hallucination, they have visual hallucinations, and so it means that the Wernicke area, which is involved in the auditory, is involved. The Broca area, which is involved in the speech, also altered, and obviously hallucination is comes from the disturb in the neural circuitry. This is actually how it looks like. This is the presynaptic neuron. This is the postsynaptic neuron. It's actually a glutaminergic neurons. And the genes NRG1 neuroregulin is present here with this binding, this one. This only shows that the presence of different genes and their receptors are also very important. Because if the protein is present but the receptor is reduced, still you would lead to have a schizophrenia. And COMT is the catecholamine methyl transferase, which actually is involved in the metabolizing of the neurotransmitters, catecholamine neurotransmitters. So any disturbance or the mutation in these genes lead to their activity or the abnormal activity, which could lead to the schizophrenia. So as I've mentioned, that neuroregulins, they are tropic factors, and they act through their ERB receptors. Usually ERB4 receptors or 3 receptors, they are also involved. So this neuroregulin is present in neurons, and involved in the neuron-glial communication. They are also involved in the myelination, which is responsible for the nerve conduction. It's very important. And it is, and usually in schizophrenic patient, it has been seen that NRG1 mRNA expression is increased, but its receptor expression is decreased. So obviously it will not perform its proper function. Recently in 2015 a paper is published in which it has been shown that NRG1 through its ERB4 receptor actually involved in the uptake of glutamate excitatory amino acid carrier proteins. So if the carrier amino protein is increased, which is obviously it's glutamate is the main excitatory neurotransmitter, so it means that the dopamine will be removed fast or from the synaptic cleft more efficiently. If it remains there, it will lead to the exocytosity and causes the desensitization of the receptor, which obviously in either case it will not give you the proper function and the neural circuitry will be disturbed. The second gene that we have identified is AKT1, is the serine-threonine protein kinases. It's also an enzyme which actually obviously involved in the neuronal excitability and regulates the synaptic plasticity. Besides this, it also involved in the regulation of GABA A receptor mediated synaptic inhibition. GABA A receptor, as you all know, that upon activation it leads to the chloride ion enters into the cell and causes the sedative effect or the anesthetic effect, or lower down the effect of the brain, calm down the brain. And in schizophrenia, it has been seen that the AKT1 expression is being low. So it means that if there is a mutation or there is less protein, so it means that the efficiency of this enzyme will be disturbed, because GABAergic is the main inhibitory neurotransmitter present in the brain, which actually regulate the stimulation or excitatory neurotransmitter effect with each other. Because all of the neurons, they actually fire together in coordination with each other. So it is very important that if dopamine receptors fire dopamine, or the glutamate, this is excitatory, so the level of glutamate should be balanced and matched with the total amount of inhibitory molecule. The COMT gene is catecholamine methyl transferase. It's an enzyme which actually metabolizes the catecholamine neurotransmitters. That's called dopamine, epinephrine, and norepinephrine. And it is believed that if its function is disturbed, which actually what happens where at codon 158 of this enzyme, codon 158, if the substitution of valine amino acid to methionine takes place, it will lead to the product that has less or reduced activity of the enzyme. So if you have a reduced activity of enzyme, it will not remove or metabolize the dopamine receptor, dopamine neurotransmitter at the synaptic cleft properly. So it will lead to the schizophrenic symptoms or any psychotic symptoms. As I've mentioned that they have, obviously schizophrenic patient has changes in the brain volume, and see the ventricles, they are large as comparative to the normal ones, and they also have the auditory cortex involved because of the auditory hallucination, and the visual cortex is also involved, and the hippocampal and especially the mesoglimbic area, especially the tegmental area, ventral tegmental area is involved, because they are all involved in the in producing the wrong and false type of hallucinations to the schizophrenic patients. So at the moment, we usually antipsychotics are available, which all reduces the dopamine levels, or in either case D2 receptors. So we have typical antipsychotics, and we have atypical antipsychotic. Atypical is a relatively new term, and it's second generation drugs, which all have a tendency to actually reduce down the excitatory D2 receptors, against D2 receptors. And but unfortunately, not only one drug is given, but the mixture of different drugs has been given, which actually gives a very heavy side effects, because they have not any, because in Pakistan we actually don't, the patient actually do not go to doctors very fast when they have fits or they have feeling something different or going on with them. They just thought that maybe some black magic has done to them. So they went to the shamans and to other medical things, which is not actually useful. So they usually do not go to psychiatrists. If they go to psychiatrists at early ages, then this disease could be prevented. So these are the drug, as I've mentioned, these are the drug which actually reduces some of them positive symptoms, and some of them they are reducing the negative symptoms. So usually schizophrenic patients, they actually start to smoke or try to take drugs, because drugs also do the same thing. They just increase the dopamine levels, or they block the dopamine transporters. So dopamine will remain there. So at least the heavy smokers or drug abuser, they just to try to inhibit the negative symptoms. They just, because in negative symptoms usually D1 receptors involved, which actually reduces the dopamine levels. So if you take cocaine or any other drug or smoking which increases dopamine level, which that leads to the reducing or lower down the negative symptom of the schizophrenia. But this is not the case, and it's just a temporary substitution. So in the PhD thesis work, my student has identified the socioeconomic factors, in which he has mentioned the marital status, education, salary bracket, symptoms, and drug abuse. So we have developed a questionnaire in which we have recorded and documented each and everything about the patient and the control ones, and then we have matched it. And we have used the different statistics values to identify whether they are positively and significantly matched with the schizophrenic patient or not. So we have identified that there are no age limit. Men and women are equally prone to have schizophrenia. So they are not like men are more prone or women are more prone to schizophrenia. They both are equally prone. Age limit is not a obstacle. So at any age it can be hit from age 15 till middle age, or at 59 or 60. And we have seen that a married woman, especially after postpartum depression, means after delivery of a childbirth, because of the estrogen levels, the fluctuation of the hormones level takes place at that time, leads to severe psychotic problems. And single men, because they lack in expressing themselves, they desocialize themselves, and they because of the social environment and less of the the salary, so probably they actually feel more depressed because of so they try to withdraw themselves from social gatherings. So we thought that single men and married women, they are significantly associated with the schizophrenia. Other one, the salary bracket, obviously the upper class, the elite class, or the rich class, they are also involved or significantly associated with schizophrenia. Whereas the drug abuse, especially tobacco and cannabis, they are significantly associated with the schizophrenia, because usually most of the patient in our study, they are either addicted with the betel nut or cannabis or with tobacco. So we have also identified the level of trace metals in schizophrenic blood. We have isolated, we've taken the blood samples from the hospitals of a schizophrenic patient, and we have identified the level of trace metals, because trace metals are equally important, and because they all of like chromium, lead, zinc, copper, iron, manganese, they are all involved in the different functions, metabolic functions of the body. They are act as a prosthetic group in many of the major enzyme, and without their presence, the enzyme and other metabolic pathways will not function properly. So that's why their levels should be regulated properly. And these levels and these trace metal can enter in your body through water, through pollution, and through the vegetables that you actually eat, if they are grown in wrong water, or the water which are contaminated or have high levels of metals. So that's why from there you can get the dust and the paint, the special paint which has lead in it. So this study, we have used inductive coupled plasma mass optical emission spectrometry. Is the type of mass spectrometry, but it is way more sensitive than mass spectrometry, and we have identified the level of trace metal in schizophrenic patient. But luckily in Pakistan, we haven't found any significant of metal in the onset of schizophrenia. So this is a relief that at least we have a good sign. Unfortunately, for the when we have genotyped the mutation that is present in 3 genes that I have identified in NRG1, COMT, and AKT1. So we have used different variants of NRG1. We have used SNP1, SNP2, and SNP8. The reason of using different variants is because the symptoms of variants of the same gene have are different. So if you have have a mutation of SNP1 or different variant of the same gene, you will have different symptoms. So according to the symptoms, it is very important that the mutation-based drugs should be developed, and which are more prone to have more specific to each and individual symptoms. Because if you have a mutation in general, then you have all type of symptoms. But if you have only one type of symptoms, then the drug or the normal drug, which has less side effects, can heal you. So we have used a genotype. We have used specific restriction enzyme. We have a DNA, isolated DNA from schizophrenic blood, and then we have used specific restriction enzymes, which recognizes the specific mutation present in the genes. And then we have used DNA sequencing, which we identified that where actually the allele or which allele is changed. So we have identified SNP1 by using MUN-I. This is a restriction enzyme. We have found that wild type allele GG and GA, which is present, which shows that we did not have any mutation in SNP1 from Pakistani patient. We have homozygous and we have heterozygous allele, GA allele, present in all the patient and in the control. So this is a very good thing. Apart from this, SNP2 also we have used a 2,1-I enzyme. This is a specifically designed restriction enzyme, which actually make a cut if you have a enzyme, if you have a mutation. So we also found homozygous GG allele at 187 base pair in all patient and in control levels. So it control people. So it means that we do not have any mutation in this gene either. This is NRG1 electropherogram from DNA sequencing, and this also shows the presence of C, which is a normal homozygous allele, and CT is a heterozygous allele. If we have TT, that it means that homozygous TT allele, so it means that it has a skip mutation in NRG1, but luckily we did not find any homozygous TT. We also use SNP8 of NRG1. This is the ID, SNP ID of NRG1, SNP8. And we have identified that SNP8 of NRG1 has CC homozygous mutation. It means that our schizophrenic patient have a mutation in NRG1 SNP8. So their symptoms could be different from SNP1 and SNP2. They are aggressive, they have mood swings, and they are either though we identified that they have master's degree, and some of them they are in the medical students. Apart from them, in SNP8 we have identified a novel gene, novel allele G. But at the moment, according to the literature survey, we have identified that it's not linked to schizophrenia because we didn't find any paper published that shows that they have G. So instead of A, we have G. So this is a novel and new mutation we have identified in Pakistani schizophrenic patient. This is the Heidi-Weinberg equilibrium. The statistical analysis that we have identified. So it's the CC allele or SNP8 has more frequency of having CC mutation if you compare it with the control, and it is significantly associated with the Pakistani schizophrenic people. The second enzyme that we have used is COMT, is catecholamine methyltransferase gene, and I have mentioned that if there is a substitution of valine to methionine at 158 codon, it will lead to severe, having schizophrenia. And we have identified that by using a specific restriction enzyme, we have found MET allele, which actually is the main culprit at 96 base pair, and WEL-MET and WEL they are present obviously in control and patient, and we have identified at 109 base pair. But this is the mutation one, and it is present at the 96 base pair. As I mentioned that it is a hereditary disease, so we have identified one family. Luckily, one family whose mother was homozygous for schizophrenia. She has MET homozygous mutation, and she transmitted this mutation to all of her siblings, and her daughters and her sons, and only one son has heterozygous mutation. We do not have any information about the father, so we thought that maybe he's either heterozygous or or a normal one. But we thought that it is heterozygous, otherwise the valine amino acid will not come. So it means that it's a differential transmission of a schizophrenic patient in COM gene, and we have identified in Pakistani population. We also identify the presence of AKT1 gene, and we find that luckily only G homozygous and only heterozygous GT is present. So no TT, which is a mutated form of this gene, is present in Pakistani schizophrenic patient. So in conclusion, I would like to say that the postpartum depression, financial status, cannabis or tobacco or Nestle are linked and significantly linked with Pakistani schizophrenic patient. Heavy metals, luckily they are not involved in schizophrenia, at least in Pakistan. Only genetic variant of NRG1, SNP8, has showed a strong correlation with schizophrenia. Apart from this, AKT1 gene is not involved, at least in Pakistan. COMT-well 158 methine means substitution of valine with methionine at 158 codon is strongly correlated with Pakistani schizophrenic patient. And tobacco can trigger COMT-well 158 MET mutation, because our patient, both patient that I have mentioned, which has a maternal with the family and the patient, they all are tobacco user, heavy tobacco user. Either they are cocaine or heavy tobacco user. In the end, these are the SNPs ID that we have submitted our gene sequences in NCBI Gene Data Bank. So these are the IDs from Pakistani schizophrenic patient. So either you can just cut and paste these links into NCBI PubMed. So they will give you all the details of the frequencies present and allelic frequencies, Heidi-Weinberg frequency, and the amount of DNA that we have used. Or either you just cut and paste this link and you will find out each and everything about the Pakistani schizophrenic patient. So at the end, I would like to thank my Professor Dr. Ata Rahman, who's actually the advisor of this university, Professor Dr. Iqbal Chaudhry, who's our director, who's here with me, and Ms. Nadira Panjwani for providing us the world-class facility, and obviously this special thanks to Dr. Iqbal, who actually forced me to come here and present, and thank you very much. And my student, PhD student, Rukhsana Nawaz, whose work I have presented here, Dr. Hina Ishtaq and other my MPhil students, my collaborators, Professor Dr. Asad Usmani, Dr. Raza and Dr. Ishaq Sarandi for their contribution of during trace metal analysis and blood sampling and the data collection, and obviously financial support from the Higher Education Commission of Pakistan. So thank you, ladies and gentlemen.
Thank you very much, Dr. Siddiqui. One question from the audience, please.
There's no one I can join in. I noticed that you said that there is no connection between heavy metals and schizophrenia patients in Pakistan. Do you know of any other data where heavy metals are involved?
Yes, heavy metals are involved in, psychotic diseases in schizophrenia. There are a lot of data. I have said that because we did not find in Pakistani population there is a link. But yes, all over the world there are heavy and so many data present that they are severely linked with the disease.
Very nice piece of
work.
With
schizophrenia and with other antipsychotic, with disease and with the psychotic diseases other that are present. Definitely, yes.
Very nice piece of work. Thank you.
Thank you.
Thank you very much. Would the presenters please all take their seats at the table over there where they have a microphone in front of them, so if the audience asks a question, you can answer.